Record. Right. So today's guest speaker is Kimberly Van Aken or Ack. She is going to talk today about Textpresso. And when I asked Kimberly for some of her basic information so I could introduce her correctly, I found out that she's also a curator. Right after her PhD, she was also a curator. So I'm kind of like, oh, a future mentor maybe, because I went from a PhD to a curator too. She's been working as a curator for Proteome, which was her first position, and then since 2003 has been working as a curator for Wormbase and Textpresso Central user. Also a curator for the Gene Ontology Consortium. And since 2015 has actually been the project lead for the annotation, Gene Annotation Consortium. So it is with my great pleasure to introduce Kimberly Van Ack to give us a great talk, or interesting talk, about the Textpresso. Thanks, Kimberly.
All right, thanks very much, Jacqueline, and thanks everybody for joining today. I know there are a few names on the participant list that I recognize, but most people here, I think, I haven't had a chance to meet. So by all means, as I go through the talk, please, feel free to interrupt with questions or put a message in the chat. I'd like this to be as interactive as possible. So I'll go ahead and share my screen here and get started with the talk. So what I'd like to do today, can everybody see that okay?
Yes, I can see it.
Okay, sort of give you some background about what Textpresso Central is and how we use it at Wormbase, and then give you some thoughts about some of the new things that we're doing with Textpresso. So let me just click. So one point just as I get started. So for a very long time, for about 15 years or more, Textpresso was just simply known as Textpresso. Excuse me, let me mute that phone. But about a year and a half ago, we published a paper on the latest version of Textpresso that has many more features, and so it was renamed Textpresso Central. So just for simplicity's sake, I will refer to it throughout the talk as Textpresso Central, but know that if you see any old papers or websites that refer to Textpresso, it's really the same thing. So Textpresso Central is it's a full-text literature search engine and a literature analysis platform that was really largely developed with an eye towards aiding biocuration. So let me, like I said, I'll talk a bit about, why and how Textpresso Central was actually created, how we use it, and where we are headed going forward. So I think all of us have probably seen slides and figures similar to this, which is a reflection of what the number of citations that have been added to Medline over the past 50 years looks like. And you can go to the NLM website and get these numbers and create this graph. But the bottom line is that the number of papers continues to increase at a rate that exceeds our capacity really to do fully manual biocuration. And so when Textpresso Central was first created at Wormbase, like I said, it was really created as part of our biocuration system or pipeline at Wormbase. And the overall goal was that we really wanted a tool that would fulfill a need for doing better targeted literature searches. So by that, we really mean what we wanted to be able to do was some semantically meaningful, deeper searches of the C. elegans literature that would allow us not just to do, say, simple keyword searches, but really interrogate the literature with specific biological questions. So in order to do that, at the time, what we recognized is that we were going to need to have a system with several key features. So we were going to be able to absolutely search the full text of papers and not just abstracts. That we were going to be able to, going to need to develop what we call categories, which are, groups of related terms that reflect particular concepts, biological concepts, that would allow us to query the literature using all the terms in those categories without necessarily having to systematically go through those terms as keywords. And then lastly, we really wanted to be able to search at the sentence level, meaning that we wanted to be able to say, search in this particular literature corpus for these types of terms, but they all have to be in a single sentence. And that's what really gives us the power to pull out the semantically meaningful search results. So the first iteration of Textpresso was published in 2004. And this is the paper in PLOS Biology here. And so at the time, it was really just for the Synovitis elegans literature. So there were about 3,800 full text articles. And the categories consisted of sort of homegrown or manually created categories, and at the time, just the gene ontology. And so there are about a little over 14,000 terms plus Go that were in the categories. And you could do sentence or document level searches, either one. And so this system served us quite well for about 15 years or so. But as we used it extensively for curation, there were a lot of things that we felt we could do to improve it. And so as of right now, 2019, we now have the next iteration of Textpresso that's called Textpresso Central. And the paper describing that is here and published in BMC Bioinformatics last year. So what does Textpresso Central now have? So again, it's based on full text articles. In addition to adding probably over 20,000 more C. elegans papers, it also includes the open access subset of articles from PubMed Central. So we now have over 1.5 million articles in Textpresso Central. The basic idea behind search is still the same. You can do keyword searches, but you can also do category searches with categories or ontologies. There are now 6 ontologies in Textpresso Central, and we have since developed a number of what we refer to as curation-specific categories. And I'll talk a little bit more later about what those are and how they get created. It's still a sentence level search, but we've now added the feature that when a user is logged into the system, and they can actually now see all the search results within the context of the full text, provided that their institution either has a subscription to the journal for that paper, or again, if it's an open access subset. And lastly, we have added the ability to actually create customized curation interfaces within the Textpresso tool, and then curators can use those curation interfaces to send their annotations out to any external curation database. Okay. So are there any questions at this point?
So, this is Lisa Harper. To make sure I understand, the papers are already there in Textpresso Central.
That's correct. Although one of the additional features that we added to Textpresso Central is the ability for users to, if they create an account, which is very simple, to upload their own sets of papers. So you can now upload PDFs and they'll go through the same processing pipeline.
Wonderful. Okay, great.
Yep.
Thank you.
Yeah. So I'm going to talk now a little bit more about actually how these papers get input into Textpresso, and I'll use the C. elegans corpus sort of as an example here, because this is certainly the one that we've used most at Wormbase. So the way that our pipeline works for this is that we actually at Wormbase do a daily automated PubMed search using just the keyword elegans. And so then what happens is that in sort of a curation form that our developer built, a curator then goes through and then sees in that form the PubMed ID, the authors, the abstract, the journal, and they just decide which of those papers are relevant to Centrobitis elegans. And then we upload all of the bibliographic data for that paper. And then we actually have one staff member at Wormbase who goes out and gets the electronic copies of all of the papers that we bring into Wormbase. So we have an electronic copy, usually PDF, of all the papers that we're going to bring into Wormbase. And then for the Textpresso pipeline, all those PDFs, and this also includes PDFs of supplemental information as well, although we're not currently able to process, say, for example, Excel or tables like that. But anything that's a PDF actually undergoes a PDF to text conversion. And as part of that conversion, then what happens is that the software then delineates sentences within each article, identifies words and phrases, marks up those words and phrases by, say, assigning them to categories. And then all of that information actually gets indexed so that when a user performs a search, they can go out, find the matching words and phrases in the search, and then return the results in their proper context of the full text. So just to explain a little bit more and give an example of exactly how this works. So here using an example of a sentence from a fairly recently published paper. So if we take this sentence here on the bottom that just says indirect immunofluorescence using an anti-flag antibody revealed that MOG LIR3 is localized in the nucleus, and then it refers to figures. So what happens during the PDF to text conversion is that the script will consult the categories that are in Textpresso and identify which of the terms in the sentences corresponds to particular categories. So I've color-coded that here for this particular sentence, and each of the different colors represents a different Textpresso category. So here again, we'll show you specifically what categories they are. So one of the things that we use Textpresso for is to identify sentences within papers that describes subcellular localization of C. elegans gene products. So we created a number of categories to do this. So we have, for example, an assay term that has things like immunofluorescence and antibody. We created a category of verbs for things like revealed and localized. We have a category of C. elegans proteins, here LIR3, the actual subcellular components such as nucleus, and this is actually distinct from the Go category. We actually use sort of a subset of terms from Go, but we also use additional terms that actually are words and phrases that people actually use in the literature. So Go doesn't necessarily capture all of that. And then we actually have a small category that really is just words like figures and tables, so that if what you really want to do when you search a paper is you want to go to those statements that actually say refer to an experimental result. This is one way that can help hone in on that. And all of this markup is actually, it's really just in XML. So if you were to go underneath the hood and look at this sentence, what you would see is the phrase immunofluorescence is just marked up in the text as, you know, an assay term like that. So I'm going to just speak a little bit more about, you know, where do all these categories and lists come from? And so we have a few different pipelines for creating and maintaining these categories in Textpresso. So one is just strictly, say, entity lists. And these are things like C. elegans gene and protein names, alleles and variations and strains, and these are all lists that we maintain at Wormbase in conjunction in part with the Center of Data's Genetics Center at University of Minnesota. And so with every Wormbase release, we would just download the latest entity lists. We also, as I said, have ontologies that are categories. And right now we have the gene ontology, the C. elegans cell and anatomy ontology, the sequence ontology, KEBI, and the phenotype and trait ontology. And we can certainly add more of those as people need them. But one of the things that we found when we were trying to use Textpresso for curation is that, you know, as incredibly useful as ontologies are for annotation and for data analysis, they're not as useful for text mining. And so we have since developed what we call these sort of customized categories that are really tailored for specific curation tasks. And we did this by actually analyzing a number of sentences from papers that described the experimental results for the data types we wanted to curate. And we just looked at the language that people use and the frequency with which particular terms and phrases were used. And then we sort of manually selected those terms that we thought would be informative for these categories. So we've done this, as I showed in the previous example, for Go cellular component curation. We also have done it for a subset of Go molecular functions, specifically catalytic activities and physical or macromolecular interactions. And we have also done it for genetic interactions as well. And this reference here describes a little bit more detail about how we did this for cellular component categories. And so categories, they're dynamic. We add more, we edit them, and that's just something that with the new Textpresso system, we hope to make that whole process a lot easier for people as well. And so this actually just shows you the Textpresso Central website. If you go to textpressocentral.org and go to the search tab, this is actually how you can perform a search and use these categories in Textpresso. So a couple of the key features on the search page. So you can specify the search scope. You can just look for words and phrases throughout a document, or you can, as I've said, look for words or phrases that have to co-occur in a given sentence. And it really just depends on what kind of search you're doing. I use them both, just depending on what kind of information I want to pull out. So you can type in a list of keywords, and the keywords can this supports a Boolean search, so you can use and or, and there's also case sensitivity. Like for example, in C. elegans, a lowercase gene name reflects the actual gene, uppercase in the literature, people are actually referring to the protein. And then if you want to search using any of these categories, you can click on this Add a Category button, and a pop-up window will appear that will allow you to browse sort of the high level of categories that we have and select one or more for your particular search query. So when I'm doing a search for subcellular localization, what I'll often do is I'll look for the 3 custom categories that we created. Again, those are things like assay terms, the actual components, the verbs, and then I'll look at the list of C. elegans proteins. And you can, like I said and showed in the example, you can add the figures and tables category as well, which is under this customized ontology. There are also additional search options. So if you want, you can filter, say, by author or journal, or you can sort your results by year. And then lastly, and this is relevant to the question about, can you upload your own papers? You can actually select the specific literature that you want to search. So typically, I will just search the C. elegans corpus, but the PubMed Central open access papers are, roughly subdivided according to topic. And then if you were to create your own corpus, and you would do that under this papers tab here, once you've done that, that would also be an option for searching, but that's tied to your login. So that particular set would only be available if you were logged in. So Megan, do you want to ask a question?
Sorry about that.
No worries.
If you guys didn't see chat, I just took a bite of my lunch. So.
Okay.
Of course.
Yeah. I was interested in the copyright status of the things you're text mining, but it sounds like most of it's coming from PubMed Central.
Yeah, so for the PubMed Central, right, we only use the open access subset, right? So those are the papers, right, that are freely available for people to text mine. So for the C. elegans corpus, though, for curation, we text mine everything that we can get at Caltech. And that's fine as far as I know, and as far as the Caltech lawyers are concerned, then if we're using that for curation, that's totally fine as well. For other external users to come in and then see, say, their text mine results in the copy in the context of full text, they would only be able to see that once they're logged in and if their institution has a subscription to that journal.
Okay, that makes sense. Thank you.
Yeah. Of course, one of the things that we would love to do in the biocuration community in general would love to do is really be able to have as much access as possible to the published literature as soon as it's published to do the text mining, but that hasn't happened yet. Okay, and so then this actually then speaks to, let me see, there's another. You're welcome. Okay, so then this actually shows you what things look like once you have searched. So if I've done the search that I showed looking for subcellular localization, the results come up and it looks like this. So for each matching paper, you see the PubMed accession, the title, and then the journal, the year it was published, the type of paper, either say a journal article or a review, a document score, which indicates the relevance of that document to your search criteria. And like I said, if you're logged in and you have permission or it's an open access paper, you would click here to actually see the results in the context of the full text. So opening up any one of these returned papers, what you'll get then is the bibliographic information for that paper. And you'll see each of the matching sentences color-coded to mark up the matching categories. And then again, if you want to see this in the context of the full text, you can just check the box and go to View Selected Paper. And then you can see both the sentence And then you can also see, in many cases, the figures as well. So that is really overall how Textpresso works, and how the searches work, how we create the corpus, and use it in our worm-based curation. Are there any other questions at this point before I shift gears just a little bit?
Yes. So this is Tanner Shan from Green Jeans. So I was wondering how much time do you spend per paper and to create a paper, and how many papers a day you get from PubMed when you type elegance?
Yeah, so our daily PubMed query of elegans probably brings in about 8 to 10 new papers a day. So compared to some organisms, that's should be quite manageable. Compared to others, that might be a lot. And then the way that we actually do our curation at Wormbase is we're split up by data type. So curators curate specific data types. And so, how much time per paper, that, of course, really varies depending on the data types. I think it would be safe to say, though, we feel like that's increasing, right, as the content of papers also seems to increase. So I can't really at this point tell you, for all the different data types, how much time the curators feel that they spend on them. But I would say we roughly get about 50 new papers a year.
Thanks.
I have a question also. I think maybe this is your next part. So if I wanted, this is Lisa Harper, I work on Maze. So if I wanted to, we don't divide up papers by data type. So if I wanted to just curate the 8 new papers that day with anything that came up, is there a way to do that?
So you could, absolutely. Yeah. So if you wanted to, I think as soon as you have an electronic copy of any papers that you're interested in, you could upload them and use them for curation. Yep.
Okay.
Yep.
And that would create my own little subset of papers.
Right.
That I get from the select literature button. Okay.
Right.
Thank you.
Any other questions?
Yeah, me. It's Tanya.
Hey.
Hi. I was wondering if you could explain a little bit on why Go terms as is are not as great for text mining as the kind of categories that you've specially curated. What's different about the terms that are in the categories that you make versus the Go terms as they exist?
Mm-hmm. Yeah. So for component, what we found is that, for example, sometimes what you might have is a sentence that said something like, immunofluorescence showed that protein X, is nuclear localized, right? And so the Go term that we would use to make an annotation from that is nucleus, but there may not necessarily even be a synonym in Go, like that nuclear localized, that phrase, would correspond to, annotating to nucleus.
So they're like word variations.
Yeah, it's word variations. It's the way that people describe experimental results.
Right.
It's even more of an issue with function. And then, as you may have noticed, we don't have categories quite yet that we use for process, where, the Go terms for process, some of the higher level Go terms, say for things like, transport or translation, we could certainly find those in text, but we're looking usually for something more specific. And then, so the standard term names that Go has come up with is great for developing an ontology. It's just not necessarily the way that researchers express those concepts or those experimental findings in the literature. So that's why we went to the trouble to create some of these curation-specific categories. And in the interest of full disclosure, I will say that when we first started using Textpresso to do component searches, I would just go in and with a particular component, try to find, proteins localized to that component, and very quickly found out that that search just wasn't specific enough for me to really pull out the kind of information that I was looking for. And so that's how we kind of got started down this road of, well, we really need to look at the sentences, how researchers express these things in their papers, and then customize our categories for searching the literature.
Got it.
I would love to see, Go and other ontologies maybe start to create a synonym category that matches what's in the literature, but that would be a big undertaking.
Yeah. And are the categories that you use, are they available as separate types of resources outside of Textpresso Central?
So formally, not that I know of, but certainly, everything we do is available for people to use. Does that answer your question?
Yeah, it does. Thanks, Kimberly.
Yep.
So I have a question. This is Effie Cannon from Maze JDB. You mentioned that you've got a web service interface. So I'm wondering if, for example, we wanted to do a bulk search for all of the gene models that we have in Maze.
Mm-hmm.
Would there be a way to do that without shutting you down, but also a way to do that?
Yeah, so one of our new developers, Valerio Arnabaldi, is working on APIs for Textpresso, and so I can put you in contact with him to talk more about the technical details of that. But in theory, I think that should be possible.
Okay, yeah, because I just did a search through your interface, and I thought, hey, this looks like this could be useful. But then, of course, we've got several thousand genes.
Right.
So,
yep.
Okay, all right, thank you.
Yeah, bottom line is, right, not all of the searches, would have to be done through the web interface.
Right. Yep.
Kimberly, we have a question on chat. There is often missed data hidden in supplemental files associated with the manuscript. Is there any special treatment for these issues?
Yeah, so as I mentioned in the beginning, so right now if the supplemental information is published as a PDF, we will convert that. But things like tables, or if there are things in, say, Word docs right now, we're not converting those. So that is an issue for sure. At this point, I don't think we have a really good technical workaround for converting and searching tables. So there are definitely some things that Textpresso cannot search through, but all of the full text, all the figure legends, all of that is available. Okay, so now I just want to talk a little bit about some of the other things that we use Textpresso to do. So one of the things that we do as part of our curation pipeline at Wormbase is that all the new papers that we come in, all the PDFs, we run them through as a very first step, a sort of document classification or triage. And this really helps us assess which papers that come in are relevant for the kind of curation we're currently doing at Wormbase, and more specifically, what data types does it contain. So two main approaches we have to document classification are we do combined keyword and category searches, and we use support vector machines as binary classifiers. So for example, the keyword and category searches, we use these to identify papers that have some relevance to human disease. So for example, we'll look through the paper for the keywords of ortholog and C. elegans, and then we'll use the categories of human or C. elegans gene and protein names, as well as a human disease category to try to pull out papers that use C. elegans as a model system for some kind of human disease. So that's one way we classify documents. The other way that we use much more are these support vector machines. And so, like I said, these are binary classifiers. So they basically take the text of a paper and say, yes, it has this data, or no, it doesn't. We trained a number of these SVMs using papers that we had already curated as positive or negative training data. We use support vector machines for 11 different data types currently, and roughly on a monthly basis, curators get email notification that here are all the new papers, and here are the papers that are both positive and negative for particular data types. And then what curators can then do is, once they get the email, they can look at the list of papers and how they were classified according to the SVM. And then we also have what we use internally at Wormbase is called our curation status form. And for every paper and every data type, depending on how we identify that data type, it will show in the form whether it was negative for the SVM or whether it has high confidence for that particular data type. And this is a really nice way for us to triage and prioritize our curation. So in the future, we're hoping that this document classification will be part of the whole Textpresso Central pipeline, and that users could come in and say, I want to do maybe these kinds of keyword and category searches, but only on the subset of papers that are positive for catalytic activity according to the SVMs. So we think doing things like that within Textpresso would make the searches even more powerful. But right now at Wormbase, these are kind of independent pipelines, although they both rely on having access to the full text. Another thing, yep.
Sorry to bust in, but I
No
wonder
worries.
if the categories that you use for worm papers, are those available on Wormbase for like your users to search on, or to identify, I don't know, to further classify papers when people are looking at them on a gene page, so that they could see, oh, this is the one in the list of 500 papers about this particular gene that was about the enzymatic characterization or something like that.
Yeah, no, we don't do that right now, but that's a really good idea. And I like that. I think we should. Yeah. What we do in Wormbase is that if the data has been curated, right, you can see that. But if there's a backlog of curation and it may not have been curated yet, we don't show that actually, we're confident this paper has, say, phenotype data. But I think that would be a nice thing to do.
Thanks.
Okay. And then, so one of the last things I want to talk about is actually community curation and how we're trying to merge Textpresso Central pipelines with our community curation pipeline. So there are a number of curation groups that ask authors of papers to help identify entities in the paper and flag data types. So Flybase, Palmbase, Terra, and Wormbase all do this. We all call it something a little bit different, but the point is that we want the authors of the papers to help us identify the data that's in their papers. So we've been doing this for about the last 10 years or so at Wormbase, but what we were doing at the time was, asking authors, to type in their gene lists, and asking authors to, check the checkbox, yes or no, it has this data type. But we really felt like we'd gotten to the point where we could change this and change our strategy. And rather than have the community actually typing things in, we could actually present them everything that we could mine from a paper using Textpresso Central, and they would just have to validate it. So we went from sort of, a data entry for the authors to more of a validation kind of system. And so what we do is we present authors with entity lists. And so they can go through, say, the list of gene names that we mined from the paper and they can add some if we miss any. They can take things out if we have false positives. And then for each of the data types, we present them, we present them with the results of the, say, the support vector machines. If it's positive, it's checked. And they can uncheck it if we got it wrong, or they can just leave it as is if it's right. And then we give them the option to provide us with more free text details about that particular data type, or in some cases, we link out to a structured data entry submission form that we have, so they can actually put in all the relevant data that we would use to curate that data type in Wormbase. So this is our new author first pass web form. This is what it looks like. So the data types are sort of arranged here, organized by section into particular widgets. So once a paper gets published, we mine the corresponding author's email from the text in the paper. We match that up with the contact information that we curate at Wormbase. And then they get an email with a link to this form. It says, help us curate your paper. And then we present them with lists. So here, for example, are the list of genes we mined from the paper, the list of species. They can go through, like I said, if we got it wrong, they can remove select ones, or if there are ones we missed, they can add from an autocomplete of Wormbase approved gene names. This is what it looks like, say, for a particular data type. So for gene model corrections or updates, we have an SVM that helps to identify papers with that data type. In this particular case, it was negative, so it's not checked. If we got it wrong, again, the authors can check it, or if it's right, they don't have to do anything. So the idea, right, is that we were sort of hoping to really provide a lot more information for authors that they can then just verify and validate. And so just a couple other things to mention about this pipeline. So like I said, we're identifying and extracting entities. We do this for genes, species, variations, strains, and transgenes in the papers. For SVMs, we identify papers with expression data, genetic, physical, and regulatory interactions, phenotypes, and variation sequence changes, gene model corrections. And then we also have some data types that we curate that we don't have an SVM for. And for these, we just include these as checkboxes that authors can just flag for us. And then we've given them, of course, an option to give us feedback, as well as a link out to the micropublication biology website, which is another project that was initiated at Wormbase, which allows people to basically publish single experimental results as micropublications. And so this is particularly great for, the data not shown in papers. So we try to encourage people to go to micropublication biology and put any additional unpublished information they might have there. And again, the nice thing about the micropublication biology platform is that it's all done with structured submission forms. So that makes the data a lot easier to get into Wormbase. It's also a great way, say, for undergraduates or summer students or rotation students who might have done some good work, but not enough for a full publication. They can use micropublication biology to get that information published and get credit for their work. Okay, so how much more time? I can go through sort of a use case of how we've integrated Textpresso Central for curation, or I can skip to our future plans.
I personally would love to hear both of those. I think if you have time, we can go over a little bit.
Okay, that's great. So what I'll do then is I'll sort of describe a use case where we have integrated Textpresso Central curation with the Noctua curation tool that has recently been developed by the Go software team at Berkeley and USC. So just a little bit of background on what Noctua is and why it came into being. So the gene ontology has typically, associated individual genes with terms from each of the three aspects of Go: function, process, and component. But it's long been recognized that these three things don't necessarily exist in isolation in biological systems, and so Go has wanted to create a more formal representation of how these different aspects relate to each other within the context of a biological process. So to do this, they developed, and particularly this is the work of Paul Thomas and Wayumi and Chris Mungle, what we call a gene ontology causal activity model. And an overview of this, I think, was recently published, or will soon be out in Nature Genetics. And so to do this, it requires some different curation processes. So they developed the Noctua curation platform to do this. So I'll just sort of walk through a certain example given some C. elegans biology. So there's a really neat developmental process in C. elegans where there are two neurons in the head of C. elegans. They are one on the left side and one on the right side, and they're sort of a stochastic binary cell fate decision that's controlled by a MAP kinase signaling pathway that dictates which of the particular of these AWC neurons will actually be able to sense a particular odorant and which one does not. Okay. So if we wanted to, say, model this particular biology in a GoCam with Noctua, we could log into the Noctua tool, and anyone who's interested can get an account to use Noctua for curation. And so what we can do is, starting in the Noctua curation tool, which is shown here, under one of the model menus, the model menus, is that we're able to then go out and link out to Textpresso Central. And when you do that as a curator, it's sort of carrying along the information of what model you're working on in Noctua. And that's important for getting the annotations to go back and forth between the two tools. But from Noctua, you can go out to Textpresso. You can perform whatever kind of search you might want to perform in Textpresso Central. And then again, viewing the results in the context of the full paper, and this is what it actually looks like. And this is showing, a lot of the markup of the paper, but also the most relevant sentences for the query. So what we've done is we've actually created a select curation form that is called Noctua here, and the curator can use that curation form to highlight specific sentences in the text here. For example, this one that's underlined that we want to use as supporting evidence for Go annotation, and then we highlight that sentence. And in the curation form, we can then fill in the annotation, the Go term, the relation between the Go term and the function term, the evidence that we're using for that, and then it will pre-populate, say, the PubMed ID, the curator, and the date. So this will link the Go annotation that the curator has made in Textpresso Central to particular text string in the paper. And then when the curator is satisfied that they have the annotation that they want, you can then send that back to Noctua. And then when you return to the Noctua graph editor, your annotation is now there. So we've done this as kind of a proof of concept that we can actually link these two tools. I think that there's a lot more work we can do to sort of make these curation interfaces a little more user-friendly. But basically, for any customized curation form, in the tab menu on Textpresso Central, in the customization tab, there's a link to create a curation form. And so this is how we did this. You can create whatever fields you want in that form. You can say, I want this field to actually use an autocomplete, say, on the gene ontology function, and it will do that. And then if you want to pre-populate the fields, like I showed, say, for reference information for curator, you have a number of different options for how you might want to pre-populate a field too. So like I said, that's sort of proof of concept. I think there's a lot more that we can do with that. Other ongoing work that we have right now. So the author first pass form that I showed you, we just launched that on the C. elegans community in early June. So we're just getting our first round of author responses to that, which we're going to do some more analysis to really see what works well, what could be improved. It's already turning out to be very interesting. One of the other things we want to do is, like I mentioned, we email authors and to let them know that, we now have your new paper. We want to have an additional point of entry in Wormbase so that we're not necessarily just relying on authors seeing an email to give us feedback on their paper. So we're going to link out to sort of more of a dashboard type interface from Wormbase person pages, so authors could do their first pass of their papers at any time. I mentioned particular data types for which we do fact extraction, for example, the Go component, a subset of function. We'd really like to expand the data types for which we do that. Particularly, we're interested in doing this for phenotypes. And I sort of mentioned our initial approach to developing categories for fact extraction, which was just to do some analysis, essentially, of word and phrase usage in sentences. We'd like to be a bit more sophisticated about how we do that. And so one of our curators, Chris Grove, has been looking into doing classification, SVM classification on a sentence level for phenotypes. So hopefully that work will be promising. And then lastly, as I said, we have a proof of concept for getting Textpresso Central to talk to an external curation forum of Noctua. Additional work I'd like to do on that form is to be able to provide more contextual information for annotations, as well as I showed you function. We'd like to be able to have component and process included as well. And then I showed you a graph editor interface for Noctua. There are other more form type interfaces, and we'd like to get those to talk to Textpresso Central as well. And overall, the goal is always to try to make the curation process more efficient for curators. And so last but most importantly, to acknowledge all the people who have contributed to Textpresso Central. It's really such an integral part of our Wormbase curation pipeline. I cannot imagine working without it. So the Textpresso Central PI is Hans-Michael Mueller, and he's been there from the beginning and done a lot of the work on bringing the original Textpresso up to speed with Textpresso Central. As I mentioned, Valerio Arnabaldi is one of our new developers, and he's been great, particularly working on the new author first pass pipeline. Valerio was just visiting Europe PMC last week, so we're in conversation with them about maybe how we can help each other. Maybe we could provide some of Textpresso's functionality at Europe PMC. So I'm really looking forward to hearing back from him about that. Daniela Ricciti is a curator at Wormbase and an editor at Micropublication Biology. She and I have worked with Valerio on the author first pass. Juan Carlos Chan is a developer at Wormbase who does a lot of our paper curation forms. Of course, Paul Sternberg, who really had the vision to get all of this going in the first place. Seth Carbon at the Gene Ontology Consortium in Berkeley for all of his help with integrating Textpresso Central, and Noctua. All the worm-based curators who give a lot of feedback and hard work to try to make the system as good as we can get it. And then lastly, a few former Textpresso and worm-based members, Ruihua Fang, who initiated the support vector machine work at Textpresso, Josh Jaffrey, who was an undergraduate who worked on the cellular component categories, and also Yuling Li, who also did a lot of work on the support vector machines as well. So thank you very much again for the opportunity to talk and for the great questions. And yeah, if there's time, I'll try to answer any more questions people might have.
That was wonderful. You're right on time. I have a question. I was sort of trying to follow along at the beginning of your talk in Textpresso Central. I'm wondering if you would ever be willing to, if some of us are interested in incorporating this into our workflow, if we could have like an online workshop where, you know, if we run into a stumbling block, you could help us, you know, okay, what do I do next here? And just so some of us could get up and running in this.
Yeah, absolutely. Absolutely. Yep. Yep. No, it's, we're always interested in bringing more people on to use the tools. And yeah, we'd be happy to do any support we can.
Well, I'm ready. So I can organize that. Would other people be interested in joining me? They're all muted.
Yeah, they're all muted. I would also be interested.
Kimberly, I wonder if there's a possibility for setting up Textpresso Central at a second or third or fourth location. In other words, if there are resources that have their own large sets of papers, and they want to apply this technology, is there kind of, here's a Docker instance or here's a box.
Yeah.
And unpack it and get it going.
Yeah. I think, again, in theory, that should be possible. This is all open source. And so I would defer the details of that to Michael and Valeria, who are both on vacation through most of August. But yeah, once they're back, absolutely. I think that's something that we could discuss with people as well.
Thanks.
Yeah, because I was thinking, well, I've got, you know, several, you know, thousands of papers on Maze I could upload, but that maybe that's not the right way to do it, to mess up your corpus. Or maybe
yeah, I don't think it would, like I said, I don't think it would mess up our corpus in any way, because certainly users can come in and select what literature they want to search. But yeah, I think it also may depend on, you know, what's easiest for the groups who want to use it.
Okay.
So I think that would be a great conversation to have with Michael and Valeria.
Okay, maybe we could get them, all three of you for an hour sometime to help us sort of walk through this.
Yep, that would be great.
But I now have an account. I'm going to start to add papers.
Great. And if you run into any problems or stumbling blocks, let me know.
Thank you.
Do you know where Textpresso Central lives? Is it on servers at Caltech, or is it somewhere in some cloud?
So I think it is now in the cloud, but don't hold me to that.
Okay.
Are there any other questions? Well, thank you so much. That was a really excellent and informative presentation. So,
Well, thanks very much for the invitation.
Yes, and thank you all for coming. And Jackie will have this recorded and up on our website, and we'll see you all next month, if not sooner.
Thank you. Bye.
So,
Thanks. Bye.
Bye.